Hello! Today in the cell biology school we look at stem cells and cell differentiation. Multicellular organisms have a ton of cells, and these cells they need to differentiate into different cell types in order to become hair, to become limbs, to become bones, to become eyes. So there is a challenge here for a multicellular organism. The challenge is to be able to divide new cells in order to replenish the body and to grow a body, but at the same time to be able to make these cells become different from each other so that they can each achieve specific tasks. So this challenge is addressed in different ways. And one of the first things to remember is that every cell of the body of a multicellular organism are coming from the same single stem cell. And these cells, they all have the same genome. They all have the same genes. So they won't be different at the genetic level. So it must be after the genetic level that this is controlled and that the differentiation occurs. So it is basically achieved by changing the way the cells read their genome. So in fact, how and which genes they will use and which genes they won't use. If we look at the stem cells and all their descendants, like here, we see that they all have, again, the same genome. Here we represented the genome with genes colored in different ways, and each of the color would give rise to a protein with a different color. This is just to best represent our cells. So let's imagine that this stem cell starts the whole thing. So let's say it is in an embryo or in a very early developing tissue. So this stem cell has the function to divide endlessly so that it can keep up with the growth of the organism. Now, when they divide, they always give rise to two daughter cells. And between the two daughter cells, then the fate will be different. One of the two daughter cells will keep the stem cell memory, so will stay with the same capacities of cell division. The other daughter cell will, in contrast, engage in the differentiation pathway, and this cell will therefore stop expressing the genes that keep the cell division capacity. But in contrast, it will start expressing different genes. So for example, here represented in yellow, and these genes will give rise to the expression of proteins that give a specificity. So let's say if it is in a muscle lineage, it would be the capacity to form muscle fibers, for example. So these cells here, they are called progenitor cells, so they can be part of a certain type of lineage, but they could eventually become many different cell types in that lineage. Again, if I take example with the muscle cells, a first progenitor cell would be called a mesenchymal cell, and it could become a cell from the fat tissue, a cell from the bone tissue, a cell from the muscle tissue. So our progenitor cell is an oligopotent cell, so it is able to become many different cell types within a certain lineage. It will still have to differentiate more in order to become a functional cell. So this progenitor cell will undergo cell division. The two cells, the two daughter cells, will eventually face different types of environment. Therefore, different types of cell-cell contacts or different types of exposures to molecules dissolved in the extracellular fluid. This exposure to a different environment will change the way the genome is read. So, for example, the cell that is exposed to a specific environment will still express the yellow gene, which is the characteristic of the lineage, but will in addition express another gene, like here, the green gene, that will produce a membrane protein, for example. And the expression of this gene combined with the expression of the previous yellow gene will create a cell that is adapted to the function it has to achieve. In contrast, the cell that is not exposed to this specific environment will remain with only the yellow gene, and because it is exposed to different environmental signals, it will express not the green gene but the pink gene, and this could be, for example, an intracellular protein that will give rise to a differentiated cell in the fat tissue instead of the muscle tissue. So you see, from the single lineage in the progenitor cell, you can give rise to two different cell types in two different tissues. So within the muscle cells, you still have different cell types, like cardiac muscles or skeletal muscles, and a fat cell could also differentiate into different types of adipocytes, like the white adipocytes or the brown adipocytes, but a muscle-oriented cell can no more become a fat cell. So their capacities to become different cell types is much more restricted than when they were progenitor cells. So we classify them by levels of potency. So the stem cell itself can become anything, so it is called totipotent. The progenitor cell is called oligopotent because it can become many different things. So oligo means many, but not all. And the precursor cell is called either unipotent or pluripotent if it still has the capacity to divide into 2 or 3 different cell types, but it is much more restricted than when it was a progenitor. So once cells are terminally differentiated, they express a selection of genes. It induces changes in shape or changes in structures. This terminal differentiation state is associated with a fully functional cell, and we can also identify it by the means of differentiation markers. So a marker is anything that can help you identify the stage of differentiation and the cell type. For instance, in our example here, a marker of differentiation could be the shape or the expression of the green protein at the cell surface, or the expression of the pink protein intracellularly. These 3 things could serve as a marker to differentiate this cell from that cell. The expression of the yellow gene is not a good differentiation marker because these 2 cells express it. So this is it for this little introduction about stem cells and cell differentiation.